Okay, so welcome back to this next video in which we are discussing methods used for investigating exocytosis. Okay, right, so we're currently in the prerequisite portion of this video, which is we're trying to get a decent understanding of how exocytosis in axon terminals is actually achieved and how it's coupled to the arrival of an action potential in the axon terminal. Okay, right. So we now have seen the formation of these trans-snare complexes, and we've seen how the progression of the zippering up of these trans-snare complexes is blocked by this clamp protein, which is complexin. Okay, and that halts the zippering up process and stops the two membranes being brought so close together that they start to fuse. Okay, now what we want to see is how when this calcium signal arrives, which is the indication that an action potential has arrived at the axon terminal, how that's going to lead to the complexin being removed from these trans-snare complexes, and that is thought to be done by the Synaptotagmin protein. Okay, so let me discuss then the structure of Synaptotagmin with you. Synaptotagmin is a protein that is present within the synaptic vesicle membrane. Okay, so once again, here is our synaptic vesicle, and synaptotagmin is going to be present within this synaptic vesicle membrane. So again, it's an integral membrane protein. This time, unlike VAMP2 or synaptobrevin2, its amino terminus is inside the lumen of the synaptic vesicle, whereas in the case of synaptobrevin2, it was the C terminus inside the lumen. Okay, then on the cytoplasmic side, you have two extremely important domains which are capable of binding to calcium. Okay, and these are a conserved domain structure that you find in many different proteins known as a C2 domain. So this first C2 domain here is known as the C2A domain of synaptotagmin, and the second C2 domain here is known as the C2B domain of synaptotagmin. So let's highlight these up. So we'll have the C2A domain of synaptotagmin in vivid purple here, and we'll have the C2B domain of synaptotagmin in turquoise here. And these domains, as I say, are capable of binding to calcium. Okay, so let me just discuss with you then the structure of a C2 domain in a little bit more detail. So basically, C2 domains are described as beta sandwich structures. Okay, so let me explain to you what a beta sandwich structure actually is. So the idea behind a beta sandwich is that you create two beta sheets and you stack those beta sheets on top of one another, and that's what a beta sandwich is. So let me show this. So here is one beta sheet here, okay, and then stacked underneath it, you'll have another beta sheet, like so. And this stacking of two beta sheets on top of one another, this is then what's called a beta sandwich. So if I make this picture a little bit more detailed, we might have the actual polypeptide going like so, forming one beta sheet there, and then it might move onto this bottom one here, like so, and form another beta sheet here. And it's these two beta sheets stacked one on top of each other that makes a beta sandwich. Okay, now, in reality, the way that the polypeptide actually moves between these two sheets will be far more complicated than what I've drawn here. Okay, so it'll be going all over the place, and these beta strands will not be in order, basically, in the way that I've drawn it nice and simply. But this gets the point across that it's two beta sheets, one stacked on top of the other. Okay, now, these structures then are capable of binding calcium in certain of these loops in between the beta strands, okay? And some of these loops are capable of coordinating a calcium ion within them, like here. Okay, so in turquoise there, that's representing a calcium ion. And loops that are capable of coordinating a calcium ion at the center here are known as calcium binding loops. So this here, this is a calcium binding loop. Okay, right. So not all of these loops will actually be calcium binding loops, but in each one of these C2 domains here, you have some calcium binding loops. And this means that C2A is capable of binding 2 calciums, and overall the C2B domain is capable of binding 3 calcium ions. Okay, so overall then, synaptotagmin is capable of binding to 5 calcium ions. Okay, now each one of those calcium ions carries a double positive charge, so overall this gives the synaptotagmin protein a plus 10 charge when these calcium ions bind to the C2 domains. Okay, right, so when the calcium signal arrives in the axon terminal cytoplasm then, these synaptotagmins are going to bind that calcium, and what then is going to happen? Well, basically, there are many things that synaptotagmin binds to. Okay, so the first things that I'm going to tell you about, they may have more importance in the fusion than I'm about to tell you. Okay, the way we're going to look at it is these first 2 things that I'm about to tell you, which synaptotagmin will bind to, are involved in just bringing it into the right position so that it can then bind to the final thing, which then is the thing which allows this complexin to break away from the trans-snare complexes. Okay, so let me tell you about the 3 things then that synaptotagmin binds to. So the first thing is that it's going to bind to acidic phospholipids. Okay, right, so what's an acidic phospholipid? So basically, an acidic phospholipid is a phospholipid that is capable of donating a proton away. Okay, so basically, the definition of an acid is any molecule that has a proton attached to it and is capable of donating that proton away, and then when it has donated that proton away, it will then have a negative charge. That's the key thing. Okay, so we can represent it like so. Here is our acid molecule, and it's capable of cleaving apart into a proton with a positive charge, and then the other molecule which will have a negative charge here. That's the key part of this, that these phospholipids can donate protons away, and then once they've donated the protons away, they will then have a negative charge. And that's the key to this, that these acidic phospholipids will have negative charges, and the plus 10 charge of synaptotagmin can bind to these negatively charged phospholipids. So the idea then is that in this synaptic vesicle membrane you will have lots of synaptotagmin proteins, and now these C2 domains might be binding to acidic phospholipids of the inner leaflet of the phospholipid bilayer of the plasma membrane. So that's one of the things that it now binds to. The second thing that I want to tell you about it binding to is what's known as PI45P2, okay? Or for short, just PIP2, so people will often just call this PIP2, okay? Strictly speaking, you should call it PI45P2. Okay, so let me tell you about PI45P2. Okay, so, in full, this name, PI45P2, what it stands for is phosphatidyl inositol 4,5-bisphosphate. So the first P is for phosphatidyl. The I is then for inositol. Okay, and then the 4,5 is for 4,5, and then the P2 is for phosphate, and then the 2 means bisphosphate. So this is phosphatidyl inositol 4,5-bisphosphate. Okay, now, this is a phospholipid. It's a quite rare phospholipid that you can find in the plasma membrane, but here's the important thing. You do absolutely not find it in the synaptic vesicle membrane. Okay, so the vesicle membrane will not have any PI45P2 in it, but the plasma membrane does have some PI45P2 in it. Okay, so let me just talk you through the structure of phosphatidyl inositol 4,5-bisphosphate. I'm just going to show it in cartoon form, okay? And looking at the structure of this will help us out later on when we come on to discuss certain in vitro fusion assays, okay, which are going to involve dyes being conjugated to phosphatidyl ethanolamine. Okay, so let's start off with the phosphatidyl prefix here. Okay, so what that means is that the molecule is going to contain a phosphatidic acid molecule, okay? It really means that the entire molecule is going to be based on the structure of phosphatidic acid, which is one of the basic units of most phospholipids that you find within the plasma membrane. Okay, so let me show you a cartoon structure for phosphatidic acid then. Okay, so this is my cartoon structure for phosphatidic acid, and I'll show you the different portions. So this vertical line that I have now coloured in green, this is representing glycerol. Okay, now glycerol is the old biochemist's name for a molecule that would now more properly be called, and I've realised I'm just writing this where I would like to extend my picture later, never mind, which would now be called propane 1,2,3 trial. Okay, and although propane 1,2,3 trial is more of a mouthful than glycerol, it's a more useful name because it tells us exactly what we're dealing with here. It tells us that we're dealing with a 3-carbon molecule, that's the propane, where you have alcohol groups coming off all 3 of the carbons, 1,2,3 trial. Okay. Now, of the alcohol group that comes off the first and the second carbon of the glycerol molecule, you are going to attach long-chain carboxylic acids, which are these horizontal lines shown in orange here. So these are representing long-chain carboxylic acid. So carboxylic acid molecules with an extremely long hydrophobic tail, basically. Okay, and these are lipid molecules, basically. They're very hydrophobic. They don't want to interact with water, and that's the characteristic of lipid molecules. Okay, so onto the first and second alcohol groups of the glycerol molecule, you will attach these very long chain carboxylic acid lipid molecules. Okay, then off the alcohol group that comes off the third carbon of the glycerol molecule, this purple group that I've shown here, this is a phosphate group, okay, attached to the alcohol group that comes off the third carbon of the glycerol molecule. Okay, so that then is the structure of phosphatidic acid, and it is a phospholipid on its own, and you do find some phosphatidic acid within the plasma membrane. Okay, so it fulfills all the characteristics that you need to be a phospholipid, and there's not many of them. All you need to fulfill in order to be a phospholipid is you need a phosphate group and a lipid group. It has 2 lipid groups and a single phosphate group, so it's certainly a phospholipid. Okay, right, so phosphatidyl inositol 4,5-bisphosphate then is going to be based on the structure of this phosphatidic acid molecule. To create phosphatidyl inositol 4,5-bisphosphate, we firstly now need to create phosphatidyl inositol. Okay, so next portion of this naming then, let's study inositol. Okay, so inositol then. Inositol, again, is an old biochemist's name for a molecule that would now more strictly be called cyclohexane 1,2,3,4,5,6 hexol. Okay, and although that's certainly a bigger mouthful than inositol, it's a more useful name, again, because it tells us exactly what inositol actually is. It tells us that it's a 6-carbon ring where all of the bonds between those 6 carbons are single bonds. That's what cyclohexane is. Okay, and every single one of the carbons in that ring has a single alcohol group coming off it. Okay, so I'm going to show inositol then quite simply as a hexagon here. Okay, like so. Okay, so that's my inositol ring there. In fact, I'm going to draw this picture out again where we've got more space. Okay, so here, those are the long chain carboxylic acids. Here's the phosphate group, and now I've attached this inositol ring, which I'm just going to represent as a hexagon. Okay, now remember, it is a hexagon of carbons, but every single one of those carbons will have an alcohol group coming off it. Okay, and basically, one of the alcohol groups that comes off one of these carbons is going to interact with the phosphate group of the phosphatidic acid molecule, and that's how you've attached this inositol molecule onto the phosphatidic acid molecule. Okay, and this structure that we've now created here, this is phosphatidyl inositol. Okay, right. Now, to turn this into a phosphatidyl inositol 4,5-bisphosphate, all we have to do is add 2 more phosphate groups off this. Okay, so this carbon here is number 1. Okay, this carbon here is number 2, number 3, number 4, so we'll stick a phosphate group off the alcohol group that comes off carbon number 4, and this carbon's number 5, and we'll stick a phosphate group coming off carbon number 5. Okay, and you might be screaming, well, why is that not called phosphatidylainositol 3,4-bisphosphate? What's the difference between this carbon and this carbon? There is a difference between this carbon and this carbon, basically. The 5th carbon and the 3rd carbon are not symmetrically identical in inositol, or at least not in the form of inositol that is used within the cell. Okay, so there's a complexity here that we haven't really gone into, which is the fact that inositol actually has a huge number of different optical isomers. Every single one of the carbons is to some degree a chiral center. Okay, at least, I don't know how quite to say it. It's not quite a chiral center in the way that you usually think of it as, but it does have optical isomerism, basically, the inositol molecule. Okay, and it actually has 9 different optical isomers. Luckily, there is only one optical isomer that's actually used in biology, which is known as myo-inositol. Okay, so if you ever hear anyone talking about myo-inositol, that just means the specific optical isomer of inositol used in biology. But in this optical isomer of inositol, the 3rd and the 5th carbon are not equivalent. They're unequivalent by a very subtle fact, which is they are optically different, basically. So it is off the 5th carbon, basically, rather than the 3rd carbon. So just know that there is a very good reason for why it is called phosphatidylainositol 4,5-bisphosphate rather than 3,4-bisphosphate. Okay, right. So this is a normal phospholipid within the plasma membrane, but not within synaptic vesicle membrane. So let me just draw it here. So here we go. Here are the long-chain carboxylic acids. Here is the phosphate group coming off the alcohol group off the 3rd carbon of the glycerol molecule. Here is the inositol ring. And here are 2 phosphate groups coming off that inositol ring. So let's colour this in. Here are the long-chain carboxylic acids. Here's the glycerol molecule. Here are the phosphate groups here in pink. And here in blue is the myo-inositol ring, or just the inositol ring. Okay, so it's basically just a phospholipid with quite a big head here sticking into the cytoplasm, but it's still a phospholipid. It still meets those essential criteria of having lipid groups and having a phosphate group. Okay, right, so that's phosphatidylainositol 4,5-bisphosphate, often abbreviated just down to PIP2, okay, PIP2. So this is present within the plasma membrane but not the synaptic vesicle membrane, and basically what can happen is the C2b domain of synaptotagmin, once it has calcium bound to it, once it has these free calciums here bound to it, it can actually bind to PIP2 within the plasma membrane. Okay, so both of these things here involve the synaptotagmin in the synaptic vesicle membrane binding to the plasma membrane, basically. Now that might serve to actually bring the 2 membranes closer together, or it might just serve to get the synaptotagmin into the right position so that it can now make the final connection that it binds to, or the connection with the final thing that it binds to. Okay, so the final thing which synaptotagmin binds to once it's bound calcium is it binds to syntaxin 1. Okay, so it binds to syntaxin 1 within this 4 alpha helix bundle. Okay, and the idea then is that when the synaptotagmin binds to the syntaxin 1 in this 4 helix bundle, that then causes conformational changes which cause the complexin molecules to cleave off. So the idea is that the synaptotagmin releases the clamp on these 4 helix bundles. They then finish the job, basically. They continue the zippering up process right till the end, and then they bring these membranes so close together that they then fuse. Okay, so now what I want to discuss is how the membranes actually fuse, because there's something important here that's going to become important later on in the fusion assays, because we're going to look at 2 different in vitro fusion assays, and there's the potential that these 2 different in vitro fusion assays can tell us something slightly different because of the way membranes fuse. Okay, so let me just discuss with you the way the membranes actually fuse together. Okay, so once again, here is the synaptic vesicle membrane, like so, now extremely close to the plasma membrane here. And we want to see how do the two membranes actually fuse together when they get really close together. Well, the idea is they initially form something known as a stalk. Okay, so let me just show this. Okay, so there's the luminal leaflet of the synaptic vesicle here, and here is the extracellular leaflet of the plasma membrane. And now the idea is that they're going to form this little stalk here, which will be a connection basically between the inner leaflet of the plasma membrane and the cytoplasmic leaflet of the synaptic vesicle membrane. Okay, so the idea is that you're going to end up with phospholipids in this little stalk, with their heads facing out into the cytoplasm, and then their hydrophobic tails facing in towards the center there. And effectively it's just a continuation of the membrane basically. Okay, like so. If these are the heads of the phospholipids here, okay, like so, what they're going to have is they're going to have the hydrophobic tails facing into the center here, and now you've just got a continuation of that basically going round like this. Okay, and that little connection that you've made between two leaflets of the membrane, okay, you've made it between the cytoplasmic leaflet of the synaptic vesicle membrane and the cytoplasmic leaflet of the plasma membrane. That's known as a stalk. Okay, so you form a little stalk in between the two membranes like so. And then the idea is that this stalk is going to spread basically. Okay, so let me show this now. So once again, here is the synaptic vesicle. Okay, here is the plasma membrane, and now the idea is that this stalk has spread hugely. Okay, and now you've just got a single lipid bilayer here between the lumen of the synaptic vesicle and the extracellular space. So to show this, here are these heads of the phospholipids here, like so, and then here are the tails of the phospholipids facing into the middle here, and this now is just one lipid bilayer here separating the lumen of the synaptic vesicle, which I'll colour in yellow here, from the extracellular fluid, which is down here, which I'll colour in a moment in orange. So this is the extracellular fluid here. Okay, so this has spread basically, and now effectively you've just got the luminal leaflet of the synaptic vesicle and the extracellular leaflet of the plasma membrane adjoining to make a single bilayer here. Okay, and then at the sides it then gets a little bit more complicated. Then you've got the phospholipids facing into the middle aspect here. So here are the heads and here are the tails, like so. Okay, and this state here, where we're halfway towards fusion, this is what's known as hemifusion. So this is called hemifusion. Okay, and then what happens is that you get a hole forming in this single lipid bilayer that separates the lumen of the synaptic vesicle from the extracellular fluid. Okay, so let me show this now. So we're going to get a little hole forming in this single lipid bilayer now. And this little hole that forms here is what's known as the fusion pore. Okay, like so. So this new hole, I've just taken this picture and made a hole in this single lipid bilayer here. This is now called the fusion pore, and this is going to now allow neurotransmitter to move through it into the extracellular space. Okay, and then the final portion of this then is that the fusion pore will extend out. And then what you'll end up with is a picture that looks like this, okay, where the synaptic vesicle membrane is just continuous with the plasma membrane. Okay, so this is the plasma membrane here, and this is the synaptic vesicle membrane. To just show this on another picture, it'll just look like this, basically. This is the plasma membrane shown now as a single line rather than as two parallel lines, and here is the synaptic vesicle membrane, and they've joined up to form a much wider fusion pore here. Okay, so basically this membrane is just sort of recoiled back basically once you've opened that hole there. Okay, right, so that's then how the plasma membrane fuses with the synaptic vesicle membrane. Okay, right, so that now is the prerequisite bit done. Let's now turn our attention onto experimental methods for investigating exocytosis pathways. Okay, so before we start on the in vitro fusion assays, I want to talk about an incredibly useful tool for anyone who's investigating snare proteins, which are the clostridial neurotoxins. Okay, so I'm wondering whether to get a new page for this. I think so. Right, so let's turn our attention now to the clostridial neurotoxins produced by clostridial species of bacterium. Okay, so clostridial neurotoxins is the next topic, and we're going to use quite a few of these clostridial neurotoxins in later experiments that we go over, so that's why I think it's important that they go first. Okay, right. So these are produced by two different species of bacteria. Okay, one is Clostridium botulinum. Clostridium botulinum. Okay, which produces botulinum toxins, which are famously used as Botox, standing for botulinum toxin. Okay, then the other important Clostridium species which produces a Clostridium neurotoxin is Clostridium tetani, which is the cause of tetanus. Okay, these are both very, very dangerous species of bacteria which have a very, very real ability to kill you. Okay, most people have been vaccinated against Clostridium tetani because it's a bacterium that's found within the soil, and there is a very real risk that you can get infected with it. Okay, Clostridium botulinum is very, very rare to ever see someone getting infected with that. Generally, it's found growing in badly preserved home-produced food, basically, particularly home-produced sausage. If you don't make sausage correctly, you can get Clostridium botulinum growing in it, and that can be very, very dangerous. Okay, and can cause botulinum poisoning. Okay, so both of these species of bacterium then, they are gram-positive bacteria, okay, and circular in shape. So to draw a little picture of them, this is my picture of Clostridium botulinum and Clostridium tetani. Okay, but the important thing as far as we're concerned is that they produce neurotoxins that work by stopping neurons from releasing neurotransmitter. And the way that they stop neurons from releasing neurotransmitter is by cleaving the snare proteins. Okay, now we're not going to go through the actual reality of which neurons they specifically target and how they get into the neurons and all of that. We're just going to go through how these are used as experimental tools and which snare proteins they specifically cut. Okay, right. So, you can find botulinum toxins and tetanus toxins. Well, there's only one tetanus toxin, but there's loads of different types of botulinum toxins, okay? And you can find a toxin that's capable of cutting each of the three snare proteins, synaptobrevin 2, SNAP25, and syntaxin 1. So let's go through each of those snare proteins in turn and see which botulinum neurotoxins and where tetanus toxin cuts these. Okay, right. So we'll start off with synaptobrevin 2. So let's say that this is the synaptic vesicle membrane here. Okay, and let's say this is synaptobrevin 2 sitting in the synaptic vesicle membrane like so. Okay, so the key thing about these toxins is they all cut the snare proteins within their snare motifs. Okay, so all of the neurotoxins that work against synaptobrevin 2 are going to cut it in this R snare motif here. So this is our synaptobrevin 2. Okay, so let's see where the different clostridial neurotoxins cut synaptobrevin 2. Okay, so we'll start at the amino terminus then. The one that cuts most near the amino terminus of the snare motif is botulinum neurotoxin F, and for short, botulinum neurotoxin is abbreviated as BO for botulinum, and then NT for neurotoxin, and then you put a dash, and then you put F, which tells you the specific type of botulinum neurotoxin that cuts there. Okay, right. Next up, after the binding site of botulinum neurotoxin F, you then have a cutting site for botulinum neurotoxin D. Okay, so this cutting site here is for botulinum neurotoxin D. Okay. Then after the cutting site for botulinum neurotoxin D, further along the snare motif here, you then have the cutting site for both botulinum neurotoxin B and also tetanus neurotoxin. Okay, so botulinum neurotoxin B and tetanus neurotoxin actually both cut synaptobrevin 2 at exactly the same site as one another, and it's reasonably near the middle of the snare motif over here. Okay, then the final one that's closest to the C-terminal of the snare motif is the botulinum neurotoxin G. It cuts synaptobrevin 2 down here. So this is botulinum neurotoxin G. Okay, right, so those are the 5 clostridial neurotoxins that are all capable of cutting synaptobrevin 2. Okay, right, let's now move on to SNAP25. So I'll draw the plasma membrane now here so that we can put in the target snares, the T snares. Okay, so here is SNAP25, which is attached to the plasma membrane by its lipid moieties, and we know that it has the 2 snare motifs, the QB and the QC snare motif here. Okay, now all of the clostridial neurotoxins which cut SNAP25 do so in the QC snare motif. Okay, so it's this second snare motif that they cut. Okay, so once again, working from amino terminus to carboxylic acid terminus, botulinum neurotoxin E cuts most near the N terminus of the QC snare motif here. So this is botulinum neurotoxin E here. Then next up, you've got the cutting site of botulinum neurotoxin A. So this is botulinum neurotoxin A. And then closest to the C-terminal, this is the cutting site of botulinum neurotoxin C. Okay, now finally, there is one botulinum neurotoxin which cuts the syntaxin 1 snare motif, and this is also botulinum neurotoxin C. So botulinum neurotoxin C not only cuts SNAP25 in its QC snare motif, but it also cuts syntaxin 1 in its QA snare motif. So I'll show this here. So here is our syntaxin 1 protein, like so. Here is the snare motif of syntaxin 1. Here is the HABC domain in orange here. And basically, botulinum neurotoxin C cuts right in the middle of the snare motif of syntaxin 1 here. So this is the cutting site of botulinum neurotoxin C. Okay, right, so those are where the different clostridial neurotoxins cut the different snare proteins, and these are extremely important. Okay, they're extremely important scientific tools. Okay, so later on we will be using botulinum neurotoxin D specifically to cut synaptobrevin 2 and to inactivate that synaptobrevin 2. But this is the reason that these neurotransmitters are so effective at blocking exocytosis, because if you stop the snare proteins forming this snare complex, the trans-snare complex, you cannot prime your synaptic vesicles for exocytosis in response to the calcium signal generated by the arrival of the action potential. Okay, right. So we'll call it there for this video, and then in the next video what we will do is we'll start with the in vitro fusion assays. Okay, so we'll look at lipid mixing assays and content mixing assays.